Hello students, this is Mamta from Department of Botany. Today we will be studying about life cycle of cycas. Cycas is a strictly dioecious plant, that is male and female organs are born on two separate plants, and the female plant bears a male cone and female plant bears a group of megasporophylls. Now coming over to the male cone is terminally sharply stalked, compact, large and oval in shape. It consists of a central axis, cone axis, around which numerous microsporophyll are spirally arranged. That is, you can see here the central axis, what it is, and these are arranged around the central, that is axis, cone axis and all. And the male terminates the growth of the tip of the male plant. So a lateral bud here later grows into, and that is takes over as the continuation of a growth of apex. So this is another speciality of this male, that is the male, it terminates and next automatically when it terminates, that is the growth of the tip of the male plant, so a lateral bud later grows and takes over the continuation of the that is a growth of the apex and all. So this is another aspect of the cycas male cone and all. And the outer covering of the male cone, that is what you see here, has a closely set sterile ends of microsporophyll. That is, the upper portion, the outer covering of the male cone here is formed by a set of sterile ends of the microsporophyll. Usually these sterile ends processing up and also that is up the curved tips. Along with the curved tips they will be present, and this is also called as apophysis. So you can explain the apophysis both in male cone and also in the microsporophyll and all. And coming over to the LS of male cone, that is nothing but the what is called as a longitudinal section of a male cone. Now coming over to the longitudinal section of the male cone, this male cone is attached to the tip of the plant body and by by a stout broad stalk, and the cone here consists of the central axis, that is what I have already shown you, the central axis with many microsporophylls. You can see here many microsporophylls are present. Each microsporophyll is attached to the cone axis. The part of the microsporophyll away from the axis is upcurved. The part of the microsporophyll that is away from the axis is upcurved and is also called as apophynous, that is physis. This is once again a third type of definition for apophysis and all. And next, coming over to the upper surface of the microsporophyll is sterile, while the lower surface of the microsporophyll is fertile. So this is another important aspect of the microsporophyll. And a single microsporophyll is woody triangular shape, more or less horizontally flattened structure. So a woody triangular, that is triangular shape structure, and it is more or less a triangular flattened structure. So this is about the the microsporophyll and all. And once again this microsporophyll is differentiated into nothing but a fertile and a sterile part, and fertile part is wedge shape, that is somewhat pointed, this one just like a razor and all, is wedge shaped and is expanded distally from the narrow point of attachment. So this is being distally, that is expanded distally from a narrow point of this one, that is mainly of attachment, and sterile part here is pointed and upcurved and called apophysis. So once again here the apophysis is being explained in contrast with the microsporophyll. So this is about the microsporophyll, and which has a apophysis and all. Now coming over to the lower surface of the microsporophyll, of the fertile part of the microsporophyll. Just now I told you that lower surface is fertile, and so this fertile part of the microsporophyll bears microsporangia. So here you can see the lower surface of the nothing but microsporophyll. This bears that is a microsporangia is there, and this is formed by a good definite sori. That is these sporophyll, they are present in the microsporangia, and these microsporangia, they are in, that is grows forming a definite story, that is nothing but sori. That is sori is what called as a single, that is a singular aspect of the microsporangia, that is a sori. And for once again for this also they have asked in exam, what is sorus? A group of sporangia is called as sorus. So this is once again regarding the sori or sorus and all, and the microsporangia are arranged in sori around the central papillae. So all these microsporangia are arranged around the nothing but central papillae and all. And each, that is microsporangium, has many layers. That is what you see over here. The microsporangium is many layered and differentiated into 3 layers. The outermost one is a thick cuticle, that is cutinized, and the remaining inner layers are thin and they are collectively known as tapetum. So that is the outermost layer of the, that is the microsporangium. It is having 3 layers. The outer one is thick and cutinized, and whereas the lower one is, that is the inner layers, what you see here, they are all red, that is thin, and they are collectively called as, and that is tapetum. So once again they have asked the question for 2 marks, what is tapetum? So you should be able to write this, that is understand the things and that is write about it. Now numerous microspore remains enclosed inside the wall of the microsporangia. So each microsporangia encloses numerous nothing but microspore and all, and these spores are formed from, that is microspore mother cells. See, that is these spores, whatever they are, that is the spores what you see here, are formed from the microspore mother cells, and this after nothing but, that is reduction division. Each spore is surrounded by outer exine and inner intine. As you all know that the microspore consists of nothing but our coats, and once again this is 3 layered consisting that is surrounded by what is called outer exine and inner intine. So this is about the microspore and all. Each microspore, that is what you see here, these are developed from the 3-celled male gametophyte. You can see here the 3-celled male gametophyte. These are the, that is microspores are formed from this area, and this is once again where, that is inside the microsporangium itself. So inside the microsporangium there is, that is you come across microspore, and this microspore developed into a 3-celled male gametophyte. So you will be finding the 3-celled male gametophyte and all. And so this is all about the microspore and all, and overall that is life cycle, that is which now we studied about LS of male cone. And one more important aspect regarding the microspore is this microspore, what you see here, it undergoes mitotic division producing small prothelial cell. So this undergoes nothing but what is called as the word that is what is called as the each microspore undergoes unequal mitotic division producing small prothelian cell and a large antheridial cell. So this once again it undergoes mitotic division, and this mitotic division, that is, you see nothing but what is called a small prothelial cell and a large antheridial cell. The antheridial cell once again that is undergoes mitotic division. The antheridial cell also undergoes mitotic division and produce a tube cell and a generative cell. That is, this undergoes nothing but what is called a tube cell, that is gives rise to a tube cell and a generative cell. And the cycas, the pollen grains, is 3-celled. So this is the reason why the cycas is having a 3-celled and all. That is, it is having a 3 cells, that is 3 layers and all. So this is all about the LS of male cone, that of cycas and all. And once again we will be studying that is megasporophyll. See, whenever they ask you for life cycle, then you have to write about the cycle, that is the plant cycles and all. And once again you will be writing about the male cone, female cone, and next then you will be writing about the ovule lastly. So you have to write this description along with the neat label, diagram and all. So now coming over to the megasporophyll, this is a female productive organ. Since there is no female cone, that is megasporophyll form a crown at the tip like a foliage leaves. So this is the speciality that is regarding the what is called that is megasporophyll. See, that is the megasporophyll, what you see here, it is a female reproductive organ, and definitely that is, since there is no male cone, megasporophyll forms nothing but a crown. These all megaspore, that is megasporophyll, they form a crown at the tip of the foliage leaves. So it forms a crown at the tip of the foliage leaves and all. And one more important aspect of this is that is crown of microsporophyll, that is crown of megasporophyll is formed each year. That is, every year you will be seeing this formation of megasporophyll, and this it is formed in nothing but acropetalous section. That is that is what you see in the stem, in the same pattern. That is, you will be seeing the nothing but acropetalous section on the stem. And one more aspect regarding this megasporophyll is crown of megasporophyll is formed that is each year. The crown of that is nothing but a megasporophyll is formed, and each megasporophyll is leaf-like structure. You can see over here, each megasporophyll is leaf-like and densely covered with brown hairs. That is megasporophyll is nothing but what we call it as the leaf-like structure and densely covered with brown hairs. So it has brown hairs over its body. And each that is megasporophyll, that is what nothing but is distinguished into a proximal petiole. That is, each megasporophyll, once again it is differentiated into a proximal petiole, a middle ovule, and nothing but that is bearing portion and an upper pinnately dissected sterile part called apophysis. So you just please concentrate on this. That is, each microsporophyle is distinguished into nothing but what is called that is what we call it as a proximal petiole. A middle ovule is there, and this ovule bearing a portion of it, and one more, that is an upper pinnately dissected sterile part called apophysis. So that is once again an upper portion, that is which is pinnately dissected, and this sterile part is called as a apophysis. And one more important aspect of this is, that is the cycas life cycle, that is the middle portion of the nothing but sporophyll bears ovule. You can see here, that is the middle portion of the just see that is figure, the middle portion here, that is it has nothing but ovule which are born in 2 rows on either side. So this is another important aspect regarding that is cycas. And once again, coming to the ovules generally, cycas ovule are generally, that is yellow or orange in color. And one more important aspect is, these yellow or orange in color, and they will be dark green, that is dark colored, either dark green and things like that. And at the same time they have a short stalk, and they are very smooth. And that is, as you know already, that is the largest ovule in the plant kingdom is the cycas ovule. So this it has to be noted. Okay, thank you.